Yep. All right. So this is a small group. So please let's make this very interactive, right? Please stop me at any point. And same with Dimitri. I'll try to see everybody here and gauge whether or not something is boring or on track or something interesting, and then we can double click on that. Here's my original agenda. It can change. So I'd like to first, again, have an introduction about DataJoint elements, which is the focus of our U24 grant, showcasing that a little bit. And with the theme of today's tutorials, I've prepared tutorial for pipeline use for electrophysiology, and maybe a demo on the MOSIC, the motion sequencing pipeline, MOSIC. And or we can do a general DataJoint tutorials, but I think that's going to be a bit less interested here, so maybe not. Tell me, we can pick, we can change. But all right, so let's do this. But maybe just after the conversation with everybody here, maybe I'd like to focus a bit more on the actual lab operations, right, in terms of data. So a bit less about writing the code and going through the tutorials. I'm sure everybody can do that. You can just go to a website and just follow instruction. That's not that hard. But maybe like the actual challenges in labs, right? Running data pipelines, how to design, deploy, integrate, those are perhaps more interesting topics. All right, so DataJoint Elements is a program that we started 5 years ago from our U24 grants to learn from the experience we had designing different kind of data pipeline for different data modalities, and then aggregate them, standardize them, and package them into modular modular Python packages that can be disseminated to. Yes, Dimitri?
That links, we refactored our website. The link now is docs.datajoint.com/help.
Oh yeah, thanks for I'll update this.
Also, your HTTPS is really good.
Right.
We got the design.
There we go. Yes. So you can go to the websites and look at all the elements, but again, let me capture this again, right? So we have each of these are DataJoint pipelines representing the schema and table design for different modalities for physiology. And including with that are processing steps. For example, the calcium imaging here actually has sequence of steps that would load the raw data, extract meta informations, run through motion correction, segmentations, trace extractions, and then store your result in some structure based on the relational data model. Same goes for the element array EFIS, focusing on NeoPixel probes and spike sorting. A lot of these are, every one of these, some has more details and larger than the others, but all should have documentation and tutorials for you to follow through. And my plan originally is to go through one of them. Just go through one of them, show everybody how it works, how to use them, not really to the very detailed implementation, but at the high level how to use one. And what's cool about that is, as Dimitri mentioned before, the idea here are these are each of these elements are modulars. These are meant to be combined and mixed together into a multi-modal data pipeline. So in this example, again, we have different elements combined together into one big pipeline for EFIS, fiber photometry, calcium imaging, pulse estimations, and operant behavior. So we use tools like deep lab cuts for pulse estimation here, Sweet2P for calcium imaging, fiber photometry, that's custom code pretty much, and EFIS that would support Spike GLX, recording for NeoPixel probes, and KiloSort for spike sorting. So each of these pipelines can run, and you can go through the tutorials and see how it runs. The examples there we have already pre-run the processing. So for the purpose of the tutorials and demos, you don't have to sit there and wait for like 30 minutes for anything to run. In the actual use case, you basically have to set it up so that you can load data from your own collections for your own data collections in a certain format that's defined here, and then trigger the pipeline would trigger as Dimitri showed before, right? So DataJoint pipelines are sequence of steps, models in relational database, but each of the steps has the actual processing code attached to it. That's kind of like the idea of this computational database, right? Data model and code goes together, executed together, and in the right sequence. All right. And our goal is to integrate, right? So DataJoint itself, the framework is, domain agnostic. So it's a general tool to design scientific data pipelines. There's nothing specific about DataJoint that is neuroscience or spike sorting. It's a tool. We try our best to work with, to enhance the integration with existing tools, right? So these are all of the open source tools and informatics resources for this grant that we're trying to integrate into data pipelines, into DataJoint, and disseminate them. NeuroPixel probes, spike sorting using kilosource, using other kind of sorting algorithm, integrating with spike interface, or 1 photon, 2 photon imaging using, miniscope. And I think most importantly, we're also supporting data publishing. So the one mode that we have today that's working pretty well for us for the past several years is data export into Neurodata Without Borders format and then publish to Dandy archive. That's the flow we have so far right now. And to be honest, it's not as straightforward, right? So there's always little things that you have to update the code, rewrite the code to convert into NWB format before you can push to Dandy. But for the most part, if you use any of the elements we have, the pathway to an NWB export is pretty straightforward. We package the code, we at least test it there. The code is already written for you, pretty much. All right. Okay. Some example I like to highlight here for different elements. So element array electrophysiology, in short, element array EFIS, we use this quite a lot. And this is a DataJoint pipeline to support NeuroPixel probes, recording using SpikeGeoX or OpenEFIS format. And we have a processing pipeline that would basically do spike sorting on this data and store the result back into DataJoint, and that can be queried and extend to your specific analysis later. Okay, and this is the pipeline structure for this. I don't know, maybe there's about 20 tables here, and each one capture an entity in this pipeline, right? So there's one table that describes the probe type. This could be the kind of NeuroPixel probes that you're running. So is it like a NeuroPixel 1.0 or is it 2.0, single shank, multi-shank, and description of the electrodes, the probe serial numbers, the probe insertion procedures, where do you insert the probes into, EFIS recording, and then start all the clustering, the spike sorting routine, or defining your parameter sets, running through it, storing the results, so the units, spike times, waveforms, quality control metrics, all of that are now captured in this one pipeline here. What do you do at the end of that? And I'll get to that later, right? So similarly, element calcium imaging. This is 2 photon, but we have a similar pipeline for 1 photon, so for miniscopes. So yeah, starting with information about like subject session. I think this echoes quite a bit with the presentation from Peter this morning about subject and session management, right? So in DataJoint, we have elements defining the schema and table structure for storing subject information and session information, but it's nowhere near as deep and as rich as what Peter showed this morning. So we're very much open to some way of collaboration here. I think this is a great effort. So right, you define subject, you define sessions, and then as you collect data, that would be creating new scans, so new entries in the scan tables, and then the pipeline is written to extract automatically meta information about the scans. So things like how many layers, how many depth layer, what's the size, what's the sampling rate, and additional informations. And then it goes through the processing routine, right? So motion corrections, segmentations, trace extractions, QC metrics, and storing the cells, the time series of the extracted trace, and all of that stored back into the database for further analysis. This is an example of that. Currently, Elements calcium imaging supports a variety of acquisition systems: scan image, scan box, the Brooker system, and different processing suite. So Suite 2P, Cayman, and Extract, and things like that. But again, right, each of these table, as you see here, has a piece of code that attached to it. So what software or what tools that's supporting is just primarily about the piece of code that's being written there. So anyone can, just improve or customize this code to integrate their own tools and analysis routines. All right, MIDI scopes, same idea. We have starting with a notion of recording, or instead of scan, we call it recording, and information about the recording, starting with the processing task. You can combine and choose different parameters and run through the processing routine. Same thing, motion correction, segmentations, trace extractions, all that, and store the result into the pipeline. Deep lab cuts, this is really cool. This is also one of the elements that's used quite a bit. What's here is that we've captured the entire, post-estimation pipeline using deep lab cuts specifically, hence the name, into DataJoint pipelines. So it starts with, you define your subject session, and then you have a model registration, model management set of tables to after you do your training, you can register, this is my model, and you can name it model A, model B, and things like that. And those are trained models. And now you can take those trained models, apply to new data, new observations. So a whole pipeline for that would basically do the inference. You can say, this is my new recording from today. I'd like to apply that with model X from yesterday, or and then that would trigger the inference pipelines, generating all the key point time series, X, Y likelihoods and all that, and store it back into a database tables for further analysis. All right, and same goes for elements face map. So this is a deep learning models to track facial features. It mimics quite a bit of the deep lab cut pipeline as well. So there's two flavors to this. There's the SVD face map, and there's the new kind of deep learning face map. And we support both. All right, so those are a few, I'm highlighting a few, I guess, more popular elements that we use a lot in different labs and different collaborations. And now I would like to do one interactive tutorial. Maybe let's do one demo, right? Instead of interactive tutorials, let's do a demo. But at least I'd like to get through one example of how you can use these open source elements and put together a pipeline for yourself. Let's do this quick. You're welcome to follow along, but maybe I'll just focus on showcasing what's the materials out there and how can you use it. So the first one I've prepared here is an element array EFIS. This is quite popular, so I thought, this is cool to show. So you can start going to GitHub DataJoint element array EFIS. This is the GitHub repository here. We have set up everything here to be using GitHub Codespace. So again, maybe show of hand, how many of you have used or know about GitHub Codespaces? All right, cool. It's a pretty powerful tool, and we use it a lot for tutorials. So you can, the instructions are all here. What I would recommend is always, you can directly start a Codespace here. Like you can click on this code right here, and normally clicking here would allow you to clone the repository or download the zip of that. Or you can go to this Codespace right here. What it is, GitHub Codespace, what it is that GitHub has provided you with cloud resources, free cloud resources, so that you can actually run code on the cloud without any setting up at all, right? So normally, as you know, GitHub is a code repository. You can go there, you can browse your code, you can look and you can edit and change, but you cannot run anything there. But with the new GitHub Codespace, I say new, it's probably 2 years old now, maybe more. You can run code now, and we prepare a lot of our tutorials so that you can actually directly run things on GitHub without having to download any code, install anything on your local laptops or anything like that. All right, so let's do this. I click this. All right, so when you first launch this, it's going to take a bit of time to set things up, maybe a few minutes actually. So instead of waiting here, let's go back to the slides. And yeah. Okay, before I start, how many people here actually work with NeuralPixels data? Or have heard of it, or know what it is? Okay, well, and this is a good topic then, yeah. All right, so NeuralPixels data, for those that are not familiar with it, NeuralPixels are these kind of probes, high density probes, that on each probe, you can have up to 960 electrode sites, for example. Some probe has multiple shank, 4 shanks, so in total like 5,000 something electrodes. And you can record hundreds of channels, 300, almost 400 channels from these probes. And they're high density. So the idea here is that you can record from multiple region of the brain, right? Multiple depth layer. You can configure your probe so that you can record from high density neural populations. And that would allow you to actually record from a lot more neurons and then enhance or allow spike sorting algorithm to extract neurons far better. So these probes, I think they're starting out maybe 4 or 5 years ago, 5 years ago when they first started to commercial and start selling, and it's basically changing the landscape here. We're collecting easily hundreds of gigabytes of data over the course of daily, and then the processing, the spike sorting becomes harder and harder. So the element array ethos work with these kind of probes, understand how to handle the metadata, extract the metadata, store the data from these probes, and perform spike sorting. So spike sortings, I'm sure if you work with NeuralPixels probes, you probably do some sort of spike sortings, right? Do you have a favorite algorithm? Kilosort? Okay, yeah. So spike sorting is basically a procedure to extract multiple processing steps to extract single neuron spiking activity from extracellular recordings from multiple channels, right? So hundreds of channels of extracellular recordings, and go through the process of thresholding, detect the spike, cluster the spikes, extract the features, cluster the spikes, and then reconstruct it, build the templates for it, and then reconstruct the spikes from different neurons and group them together. So at the end of that, right, you're recording from hundreds of channels, and these, from the raw data point of view, you have these time series of data over, let's just say, 384 channels. You go through the entire spike sorting routine, what you ended up with is, let's just say 100 neurons, 100 units. And some units are good neurons, some units are multi-unit activity, some units are just flat out noise. But then from each of those units, you get the spike times. So essentially the time of the action potential for that over the course of your recording. And then you can start doing different kind of analysis on top of that. You can align that to, for example, your experiment events of interest, right? Maybe you have a stimulus, maybe the animal responds to something, and then you can align the spikes and kind of see what's the neural population is responding to external stimuli. Okay, let's go back to here and see if it's done. Still building. Okay. Maybe now is a good time for me to actually show the pipeline on the platform. So I've basically pre-created that same elements array ethos pipeline on the DataJoint platform, and this is a way for, at least for me, to interactively play around with this pipeline, explore this pipeline, right? So, so maybe I can expand the high-level structure of this pipelines. So we start with every oval here is a schema, and a schema is a group of related tables in DataJoint. So when I use the word tables here, these are all database tables. So we start with, the data start flowing from lab informations, and then subject information, and then session informations, and then we get into ethos, and upstream from ethos, there's a few more tables describing the probe. So for example, let's click here. In this element array ethos, probes are defined by these 5 tables. There's probe types. So a probe type, let's see if I have any data in here. Yeah, so as you can see, probe types here are just the type of the probe, right? So these could be NeuralPixel 2.0 multi-shank or single shank. And then for each of the probe types, there's a table defining the electrodes information. So this is capturing the geometry of this probe, right? So information such as the electrode number, XY coordinates, and the shank number, and things like that. Once you define your probe, when you actually start the experiment, this is the kind of like the heavy lifting part of that. So the EFIS elements, EFIS pipeline described a sequence of steps for you to capture your EFIS recording. So start with a session. In one session, you could do multiple EFIS recording. For example, if you use 4 probes, then that would be 4 EFIS recording, 1 for each probe. You can do, and then each of those EFIS recording would have a certain channel mappings that goes with that. And these meta information are typically easily forgotten or not well captured. And if these are not captured well, and we work with many labs and projects that are struggling with this channel mapping information here. Without this important channel mapping information, you cannot do any meaningful spike sorting, or at least like the standard ones. So these at least define the baseline for data modeling and data structure that you need to gather your meta information for before you can actually run this EFIS pipeline. So what's next after that? So the next steps would be to create a clustering task. So a clustering task is basically saying that I want to apply kilosort 2.5 algorithm to this recording, and and then the rest of that would basically be triggering that kilosort processing, or maybe a few steps before that. So pre-processing step, right? Filtering, remove bad channels, common average reference, filtering, and then you actually go to spike sorting. You run through the spike sorting. Depending on how long the data is, it could be hours of time, right? So maybe 3 hours, 2 hours, 3 hours that would take to run through the spike sorting. And here's where I'm actually sharing a bit about the actual day-to-day labs operations here, right? So the tricky part about these steps is that every single step here requires different hardware infrastructure. So some steps require a lot of CPU and RAMs, and a few steps, just one in this particular pipeline, require GPU. So you have to set up your infrastructure in such a way that can support these different sequence of steps, different steps, right? So, I mean, one simple solution is just to buy like a massively powerful machine with GPU and just run everything there. Sure, I mean, that works, but essentially you kind of like waste a lot of resources running very little things on a very powerful machine. So we break these tables out into, the steps out into different tables, and then with DataJoint we can actually attach different hardware specification to each table. So you can say something like, this table should run on this machine, the other table should run on the other machine. So you can say, all right, the expensive processing step run on the expensive GPU machine. The cheap ones run on free resources or something like that. So you can configure it that way, and then to kind of like optimize cost or performance. All right, and then after you go through the different steps here, like clustering, extracting waveform, calculating quality metrics, the results of all of that are stored in database, back into DataJoint, and from there you can make iterative queries to get down to your session of interest, or even electrodes of interest, units of interest, and fetch back the spike times, for example, or waveforms. All right, so let's see if it's done. All right, this is done here. So if you want to go through the tutorials online, in short, you can follow the instruction in the README here. The one step you actually have to do is to actually deploy a MySQL database, and it's captured in the README in the instructions, so you can follow that. What I'm gonna do here is and there's one notebook, and it's called tutorial. You can click to this notebook, select a Python kernel, and just run through these steps here. I'm gonna run through these very quickly. Please stop me if you have any questions. I want to do this at least once so that you can understand what it takes to use existing open source DataJoint elements and start putting them together into your own studies, right? So, and maybe I can show the code, the actual code a little bit. So this is the DataJoint code that describe that EFIS recording table. So the eFish recording table has these attributes right here, and maybe I can switch back to, oh, not this one. Which one? Yeah, this one. And I can actually go to the eFish recording table and show you what the data would actually look like being stored in the in that table, right? So you store information about like subject name, session ID, insertion number, electro configurations, acquisition software here, sampling rates, recording durations, time, and things like that. So the code is actually captured here, and you can actually read the code here. And more importantly, as we said, right, DataJoint is about computational database. So every step of the way has a function, we call it a MEC function, that would basically capture the transformations and the generation of the data into that table. So you can read all of this here, and I can explain really quickly what this piece of code does. All it's doing is it's going to go upstream to the table upstream. So sessions is we'll find out where the actual data is at, open up the files, load the data, parse whatever, extract meta information about what probe is used here, what's the sampling rates, recording duration, time, things like that, right? So the actual code is being written here, and you can actually read it. And there's actually one cool thing I'm gonna try in a little bit. You can see on my right hand, there's this Ask Copilot. I tried yesterday. It's actually pretty cool. You can ask the Copilot about this pipeline. It knows pretty well. It understands how to read this pretty well. Right. So same goes for the local field potentials table, LFP. The definition is here. So we capture for each eFish recording, we're capturing the mean LFP, and then we're capturing the LFP on a per channel basis. This is being stored here. And again, there is this one MEC function that's actually capture the code that would go to the files, open up the file, extract the data, maybe apply some filtering, and then load the results, store the results. That's kind of like what this big MEC function is doing. Okay. Okay, let's run through this. So, all right. So first, we described quite a bit more here about what these elements are. If it's basically all that I've just described, and we also described how to use this with any other elements if you want to, right? How to use like basically Lego pieces, connect them together. Connect here, and then I'm just gonna run through all the pipelines real quick. Okay. So in DataJoint, there's a function called diagram. You can actually plot or display the entire diagram of your pipeline. And I mean, all right, this is quite massive, right? There's a lot of different tables and how they're connected to each other to put together this different sequence of steps. I mean, each table is meaningful here to capture how the data are stored, what transformation, what processing steps, what analysis routine is being applied at each step, and what data are stored in each steps. The way you work with this pipeline is there's first to start with a subject table. So you need to, here is describe the subject. But the first thing you need to do is you need to create a subject or insert a subject. So this is where DataJoint has this insert one functions to basically insert a row in your database. And then you can show, so as you can see, I've just inserted subject 5 here. And then the next thing is the session table. So the way you insert entries into database, into DataJoint is creating a dictionary. It can be a dictionary or a string. In this, I'm showing here inserting using a dictionary, Python dictionary. So create a session and then insert a sessions and showing it. And then this is also where you specify where the data is. So this is important. If you want to use this locally, you have to set up where to find the data, right? So in this case, you would expect to find the data for this subject and session, this particular session in this location on your local file system, for example. And then you insert the probe. So as you can kind of see, right, we're following the very similar procedure as Peter was showing this morning, right? When you create meta information, there's a certain sequence of steps that you follow. So there cannot be an eFIS recording before there's a subject or a session, right? So you follow that sequence of steps. And this is where I think integration points are very nice with any other electronic lab notebooks or quality management system. Right. Okay, so create probe insertions. All right, so now that all the metadata are being captured, the next set of tables are empty, but this is where DataJoint comes into place, right? So what I'm running here is the populate function. So what happened here is that all the metadata has been defined. Now it's time to actually run the code. And running the code, running that MEC function that I was just describing is pretty as straightforward as running .populate here. So this is the name of the table, eFIS recording .populate. It will run something successful. I probably should have cleared all the notebook outputs first. So there's now an eFIS recording. And then we create the task, the clustering task, and this is important. So if you want to, so this Elements Array eFIS support Spike interface. How many of you are familiar with Spike interface? You use it? Great, yeah. So, oh, that's awesome. So we support Spike sorting using Spike interface. It's actually quite nice. And the way we define that is that you can define parameters that are Spike interface compatible, and you can define it similar in this way, right? So if you notice, all of these parameters are kilosource specific. These parameters are Spike interface specific as to what they do post-processing. You can create a parameter set, and then you can create a clustering task. Basically, a clustering task is basically just saying that I want to apply this parameter set to this eFIS recording. And then you just run through all of your populate routines. The rest of this is just, DataJoint will actually execute the MEC functions, the code, processing all of that. Sometimes it would trigger Spike sorting, and it's probably gonna take 3 hours or so. What we've done here is we have pre-calculated the results, so we actually already run Spike sorting on this, so we're just pretend that's happens already, and then just load the results back in. All right. So once you have that done, and it's still actually taking a bit of time here, right? So as you can see, it's running. After that, you can visualize the results. So now your basic eFIS pre-processing pipeline is done. You have units, you have spike times, you have waveforms, and a number of quality metrics to check. So you can now query the database and fetch the results and plot the results. I guess one thing I would highlight about DataJoint is that in DataJoint you actually store the time series as well. You can also store scientific data types. So you can store images, you can store NumPy arrays, you can store Python dictionaries. So it's provide a lot more flexibility as to what you can store there. So if you insert a NumPy arrays, you get that exact same thing back, and then you can fetch and plot and all that. Okay, and so as you can see, I'm plotting the raster plot here, sort of, and also what else? Okay, the waveform, for example. Okay, I want to do a bit of yes, Dimitri.
Yeah, so this is an example of how you run analysis with just open source code. This is just have you seen the presentation I presented yesterday for Seek? Like how it works now with the platform.
Yeah.
So this is more if you don't have any of the GUIs of the platform. This is how you would execute just the open source code.
Yep.
I think part of what we want to show is with the platform, once you set it up, just how that process is being run.
Yeah, perfect. So I think that's a great segue into that. So let me switch over to that and then show that, right? So in short, right, so this is the open source routine. You can always run this, and as Dimitri mentioned, right, people are start building out their own external tools around this, right? So they build ELNs on top of this, they build dashboard, GUI, and they build orchestration routines. So instead of having to run, manually go and run .populate, they added something or they invented something. This is our approach to all of that. So let me switch over to, and this is probably a good example to use to look at the element mosaic. So what you're seeing here is our initial implementations of elements mosaic on our platform. So again, there's still that open source elements mosaic. that you can go through the tutorial, same thing. But what we have done here is, let me show this first. So first I am I'm showing the overall pipeline here. So this is the overall architecture of this Mosaic routine, right? So you first start with subject and sessions, and then you're doing some sort of key points detection, so post-estimation. So here we use deep lab cuts as an example. There's a training of the deep lab cuts model, and then there's the inference step, right? Applying the models, manage the models, apply the models. After that, you go to the Mosaic side of things, where you first train your Mosaic models. And these are the tables associated with the training steps. So this is Mosaic train, and as you can kind of follow down the tree here, you can follow step by step the different steps to go through the training routine. And one thing that I like to highlight here is the color code for these tables. Green means manual tables. So the important thing is that every time you see these manual tables, that means there's a manual steps in the pipeline. The user have to go somewhere to make a decision. For example, right here, they need to create a PCA task, a principal component analysis task, where they would have to select which body parts they want to run this PCA on, and the output locations, right, directory. And then the pipeline, once this is created, the pipeline would automatically get triggers to run the next two steps. So this is the preparation for the PCA and then the actual principal component analysis right here. Once that's done, there are the two next steps here. And please stop me if there's a sense I'm flying through this real quick, if you want to have any more deeper questions into it. Yes?
It's not a deep question, it's just we cannot log in to follow this. We need an account to...
Yeah, yes. You do need to have an account to log in to kind of like see things right here. But so as you can see, like any pipeline, right, will have manual steps along the way. It's hard to have a fully automated pipeline. We can make that happen, but in many cases, you want to have like checkpoints in here, right? Visual inspections of the results, curation, or something like that. And so there has to be a way to interacting with the pipeline. And of course, you can always interact with through the API, so using the DataJoint Python or using any other API tools to insert or fetch data. But people also built dashboard around it, and we do too. So here's what we've created. It's very much work in progress, but this is a DataJoint aware dashboard for this element Mosaic. What I mean by that is that the idea here is that we want to give the researcher the tool to create, flexible, customizable dashboards for their things, right? So there are, and this is our work. This is the work from Milagros, from our team. We created a number of sequence of manual data entry steps, right? So there's a first step right here, subject and session entry. You click here. There's a form to enter a new subject. Then there's a form to enter new sessions. And then when you want to start doing the Mosaic training, there's a sequence of manual data entry that user can just follow through this, right? So you don't need to really, with this kind of approach, you don't really need to be that much of a programmer. You need to define the sequence of steps and the right form for that, or the right manual entry form or dashboard for it. But the cool things about this is that everything defined here is DataJoint aware. So just from the knowledge of the table definitions, you create these forms very easily. You don't have to write anything from scratch here, right? We have open source tools that understand DataJoint structure and quickly create these forms based on the data definition, the table definition of the different tables, right? So you basically just need to design your pipelines, and then these forms can be auto-created almost. So you first create key point sets, do the body part, and this is the PCA task, and it understands the foreign keys relationship, right? For example, there's only one. This is a drop-down because PCA task is downstream from I believe body part. So there's only a few things that you can click here. In the data here, there's only one choice that you can click here. So after going through all of these kind of manual workflow, let me switch back to the pipeline. So as manual data are appearing in any upstream tables, any of the computed tables, any table that has code will automatically get triggers. So that's kind of like the workflow orchestration aspect that we've also deployed and operate on the platform as well. What that means is that we basically configure for each table here the right hardware resources to be able to run that table. So we containerize the tables, the relevant tables, all the relevant Python packages, libraries install, CUDA version, things like that, all containerized, and then the right compute resources, for example, how much RAM, would it need GPU or not, and all of those are configured already here. So whenever there's new data appearing, the jobs are just going to get triggered. So there's a what we call worker manager, is basically just track for any new data appearing. Once the data appear, we deploy relevant workers that would then go and work on the tables for that new data, all the way until there's nothing else to work on. And when there's nothing else to work on, that could means it's all complete, right? So now it's ready for the user to go and look at it. Or it hits a manual tables when the user actually have to go in there and say, oh, I need to pick a task, or I need to select a parameter, or something like that, right? The moment, once the user enter that information, the pipeline would then get picked up again, automatically finish up the rest of the steps, right? It infer the computational dependencies based on these pipelines. So essentially this tree right here is just kind of go top down. Every time there's a table with computed table or table with code that has data to be processed, we'll trigger a jobs, launch a workers or a server somewhere, run the jobs, and terminate that server. And we are doing this also in a hybrid model where, when I say the server somewhere, it could be your laptop, right? So your laptops run this thing and then put the result back into the pipelines. Right. So what else? At the end of that, and then of course the results from us scientists, we want to look at some figures, right? Some plots, something to visually inspect the results or to make sense of the results. And the pipeline itself, many of these open source tools already have like plenty of visualization resources, right? You can generate plenty of figures. If you use Diplop cuts, they have their own figure generations. If you use Spike interface, they all have that. What we also do here is to make the plotting process, well, not exactly the plotting process, but the rendering of the plot easier. So for example, we create this tab right here that has, what is this? The result for the mosaic syllables. So again, right, mosaic motion sequencing is an algorithm to detect behavioral motive, right, over the course of time. So, and it's called syllables. In this particular session right here, so for example, this subject 1, this session 1, this model 1, recording 1, there are 36 syllables found. And this is what syllables number 1 looks like, right? So this is sort of like, this is syllables number 1, this is the actual top view or a view of the animals when the animals is behaving in this behavioral motives. I don't know what this is this what? Turning or something, but these, after you have these syllables, you can render the results here. You can, these figures right here, these are outputs of mosaic pipelines. So what we do is we basically just create a DataJoint tables and then insert that PNG or MP4 or whatever data in there in the tables. And then we can create a dashboard that would visualize this pretty straightforwardly, right? So again, the goal is to make everything sort of DataJoint aware so you can design these dashboards quite easily. Yeah, so what's next from this pipeline? So the operation itself is basically user would go about data collections. Once they've collected data, they would either deposit that data into a data location that could mean to the cloud for our case, or it could be to a network attached storage or local file system, and then they can create a new session. The moment they create a new session, the pipeline would then just get triggered, right? The rest of the pipeline would just go step by step, applying the inference and then generate this set of results right here. All right, let me take a quick pause here. And any thought, questions? Yes.
So next step for a user often would be to try to refine the results. Like here you can see a lot of behavior segments have been misclassified. There are, for example, several trials that have been concatenated and just the shift, from one frame to the other
Mhm.
was misclassified because the jump of, one frame to the other suddenly was, classified as mouse turning, this kind of stuff.
Yeah.
So how would you go about refining and running again, with different parameters?
Exactly. So then that's when you go back to the train. So there's, if you remember the Mosaic here, let me show you. The Mosaic pipeline has two distinct parts of it. There's a train portion and there's an infer portion. What you've looked at are results from the inference part, right? So, model inference. So what you've looked at is like basically new video going through, being applied at models and then resulting in those syllables. And obviously it looks pretty, a bit off there. What you would do is go back to the train, right? Put more data there and try to fine tune the parameters, rerun the training. When I say rerun the training, maybe that's the wrong word. Run a new training that would generate a new model. Once you have a new models, you would reapply, you apply the new models back to that same, videos recording, and then let's see how's the results, if the results is better or not.
And how do you show, how do you access this new, what is the interface to do that?
Yeah, okay. To do what? To access the new model or
if
to
you
make
have
it?
that single, I don't know, you did that single spice sorting, you did that single run of this pipeline. Where do you see it? Like how you,
Oh, okay,
is
good.
it for the user to retrieve
So you can always interact directly with the pipeline using DataJoint itself, right? That's like, completely free. You can do whatever you want. You can write your own queries. If there's a particular view that you want to show here, you can create, you actually would have to write a bit of code to create like a new page. But the code you write, it can be pretty lightweight because it's understand DataJoint format and syntax. So as long as you can say, oh, I'm gonna store that result in this table, and then I want to create a new page like this that would just fetch from that table and play the video here, that could be straightforward. But maybe I missed your question a little bit there. But here's what it may look like. So this is the form to create a new inference task. So you basically have to select which subject I want to do, which session, which recording, all that, and then at the end right here, you pick the models. So this is critical. So here there are two models that has been trained. I don't know how good the models are. So model number 1 and model number 2, but you can name it like Superstar model 6 or something like that. And then here you would pick, that's new video recording. I'm gonna pick model number 6, and that's my good one. And then you basically submit that, would create an entry in the database, and then would trigger the rest of the job, the rest of the model inference task or tables in the database, which would then sum up that steps. The end of that step would generate the figures, put the figures into the database, and then it will show up again here, right? It will show up as new entry here. So you can, maybe somewhere you would have model number 2, and then you click that, you can compare, model number 1, model number 2, how's that gonna look like? Yeah.
Can you filter this way?
Yes, you can, let me see. Equals, well, there's only one, so there's not much I can. Yeah, but I can, there's only one here, so that's not much to compare. But yeah, that's kind of like the idea, right? Any other questions?
I just want to point out that a little bit of advertising. So Barbara here is setting up or has set up a behavior board, and we're working with her to operate this pipeline from her lab or from her board, so that all the other labs can use this already pre-set up from Harvard. And Harvard can accept, maybe Barbara can tell me. And labs from MIT or Boston University can come and process the data for the pipeline.
Right, so we are in very early stages of just discussing this, but yes, in terms of just practical uses of the core, anybody can come in and use the equipment, send your mice in, and everything that we have, so to users from any institution. But this is all something, again, very new that we are starting to get into, as you said, trying to offer as a service. So those of us, again, without a lot of coding experience, and we were just looking for a way to make it more, I think, very user-friendly, and this looks like a perfect way to kind of get into that and offer this as a service.
Yeah, so I would say, DataJoint is a bit, you have to be a certain programming expertise to use DataJoint well. So that certainly is sort of a requirement with DataJoint to be like, make the most use of that. But for that reason, there's many of labs and collaboration out there that have create tools on top of DataJoint to make the process much more user-friendly, easier, right? So this is not the only ones. I know that, Princeton has their own way of doing jobs orchestrations on Slurm, so, which is pretty awesome. And there's other GUIs that on top of DataJoint. This is the one approach that we're pushing forward here is to make DataJoint creating dashboard and visualization, plots, figures very easy because DataJoint already provide a lot of functionality there. You can store all kind of scientific data inside DataJoint, so even attachment to movies, to files. Yeah, right here. Actually, behind the scene, I believe each of these, this whole thing is this one is maybe MP4, and this one is a GIF right here. And what else can I show? Oh yeah, this is clearly just a PNG, but yeah, go ahead.
One of the participants this morning asked about integration with soft mouse. Is this something you guys have? Or like how do you handle batch loading up?
Oh yeah. That's a fun but challenging topic. So we do a lot of integration with other limb systems. So Pirates, Transnetics. The Transnetics thing is sort of ongoing MLIM system. So there's a number of that. And our approach is basically very similar to what you've mentioned this morning, right? So basically script that do the synchronization on, say, every day, every morning, right? It's tricky because we need to understand their APIs very well. Some tools have very well documented APIs, some just do not, and we just have to like figure it out. But yeah, so their APIs, we need to like do a bit of figuring out. The DataJoint APIs is well defined, so nowadays I just ask the coding agent to like just write the code for me in short, because I mean the structure is very well defined already. Like get from this table and insert into that table. It know how to figure out the rest. But yeah, in short, you do have to, at least our approach is that there's basically that script, that synchronization routines. In some cases when it gets more challenging would be updates. It's very hard to solve that problem. So, for example, we can download the new data and then add it. For example, we can upload to brand stems, right? But it's hard to detect if the original soft mouse, for example, if the sex of the animal suddenly changed. You wouldn't know how to, there's no trigger points there. So now you're that, and then in brand stem, there's always going to be a mismatch until you go and do like a diff and then fix that. But then that's a very expensive operation. So yeah, it's challenging to solve, but that's how we approach it. So we did that we are actually still doing that pretty well. We do synchronization on a Pirate, on a triggers. So user can request, hey, it's outdated, I want to trigger a refresh, or just every day, or every 4 hours, yeah. Okay, so we're almost out of time, but maybe the last thing I want to highlight is the operational aspect of this, right? So when we run through a few sessions, we can kind of like easily debug and troubleshoot and things like that. It gets trickier and more fun when you run through hundreds of sessions every day, right? And then each of these has 6 or 8 steps, one of which requires some sort of GPU and something like that. And then you suddenly have like 20 jobs that just air out. So that whole aspect of operation is like a big thing that may be a bit overlooked or underappreciated. So typically in your lab, you probably have some dedicated software engineer or data engineer to handle that, or not, or you just each postdoc or each PhD just man the station for things like that. But then they have to have extensive understanding into each steps of the pipeline. So we also try to tackle that problem by layering on top of the core DataJoint framework a layer of workflow orchestration, jobs orchestration. That's basically when I was saying that there's a way we configure hardware requirements and software requirements for each table, and then there's a trigger point that would say, hey, there's new jobs there. Go and deploy that server, run the jobs, and then report back if it's successful or errors, and then shut it down, something like that, right? So I don't know if we have it here, but I mean this is, yeah. Oh, I don't have permission here. Yeah, I don't have permission. Maybe I can pick another one. What else can I pick? Maybe this one. Okay.
Something remarkable, so basically, we can switch between so many projects, so Thinh works on a whole bunch of projects, but you can switch between them and make sense of them very quickly. And this is something that's very difficult. So that interoperability is part of what you're getting using a framework like this.
Yeah.
Would it be possible for you to be on 15 different projects and still keep up with what's going on?
Yeah, that's what I have to try to do every day. but yeah, I mean, it helps, right? So like context switching and task switching is always a hidden cost, but it sort of helps. But just look at the pipelines, and sometimes there's some errors that are quite obvious that I can just go here and track it down. Oh, the spectrogram, something went wrong, and look at the jobs. Oh, and then it turns out that some data is missing there, or there's a typo or stupid bugs that I made in the code. It will just show up right there and just change the code. But yeah. Yeah, that's just a bit more of the operation aspect of things. When the data volume gets more, right, and get larger. Yeah. All right, that's all I have. Thanks everyone. Any last questions or any thoughts? Maybe for 5 more minutes before I have to run to the airport. Yeah, thanks.
Changes I have when people make changes to the pipelines, taking parameters? How does that show up in the usual parameters?
Yeah, so not changes, but new parameters. That's how we do it, right? So if people need to play around with different parameters, it will show up as a new parameters. And do I have it here? And say parameters 104, 105, 106, for example. So it will not, there's a kind of like a backdoor way to just live update the parameters. Would we just completely, not recommend that at all. So every new parameter would show up as a new entries. And then once that new entries is there, user have a choice of say, I want to rerun that session with the new parameters. And it will create basically a new new set of entries in the tables. So you can play around with fields 2.5 and 3 and then look at all the spikes and see if they're the same or not, something like that. Yeah. So yeah, very much discouraging update on the fly. But we understand that sometimes it's important to be able to do that. So there's a way to actually sort of like backdoor it. Yeah. Yeah.
If I understood correctly, all this can be integrated with a variety of data storage, like cloud or local,
Yeah.
whatever.
So what we have here is S3, but the way you configure the project is that, at least the way I do that, is the data is on S3, but if I need to run things locally, what the pipeline would do is go to my local machine at that location. If it's not found, go to S3, download it, and then run it there. So there's a bit of data movement there, and we're trying to solve that problem smarter. But even right now, yes, you can do that. And then you can change your data storage from S3 back to somewhere else, right? Maybe local file system or switch to Google object quite easily. It's a configuration in DataJoint. But you do need to actually physically move those data somewhere, right? So if your data is already on S3 and you change the configuration in DataJoint, you actually physically move the data. Yeah. So yes.
So let's say someone is doing some type of analysis that might have been implemented in different algorithms, right? Different variants of the same algorithm.
Yes.
How does this show up in this structure, let's say?
Yeah, I think the perfect one is going back to the Julia set example that Dimitri was having. So here, there's this table right here, denoise method. You can think of your example as that would be a separate table that you keep track of, variation of algorithm or different parameter sets for the same algorithm. And so this Julia denoise table here is basically, as you can see the dependencies here, it's from both the actual image and the method. So as you have more methods coming in, technically speaking, all of your existing Julia image can get recomputed with that method. right? So that's how you manage that in DataJoint here. Of course, you can say, and the integrity is fully managed there. For example, the moment you decide this method is pretty bad, I'm going to delete that. That method, once you delete that method, all of the results from that method is also wiped in DataJoint as well. So you will not have a result with an orphan.
Well, let's say like, if your denoise method, use different variable, numbers, whatever, then you have like you have only like one denoise method. Right?
Oh, in that case, you would create. Oh, I see.
You create like multiple of these things.
Yeah.
We're like not to get like too crazy. Because,
Yeah, the approach that we take here is, let's go back to the EFIS pipeline. So we do a two-step method there. So where is it? Here. So there's a clustering method, which is like kilosort 2, kilosort 5, and so on. And then there's a clustering parameter set, which is like, all right, this is parameter set 0, whatever that means.
Okay.
Right, there's descriptions and then the actual parameters to it.
Yeah.
And this is from kilosort 2.5. And what that...
So it does say which one. So it says like, that's for 2.5, for example.
Yes.
That specific number.
Yeah, this is like 0 here. I don't know what 0 means. 0 is the default parameter set for 2.0, I guess. Yeah. So,
Okay.
right. So then you can create 0, 1, 2, 3, 4, 5, and then you can just write your notes in there. But then the actual parameters, which is here, this one right here is a Python dictionary capturing all the parameters.
Yeah.
Yeah, all the little things, right? Like the threshold values or things like that.
Okay, good.
And then what happens is when you actually run your clustering or your result, I don't have any results here, so I cannot show, but it would show up as a parameter set index here. So you would have like subject 1, session 1, insertion 1, parameter set 0, 1, 2, 3, 4, and so on and so forth. And then you can compare the result that way. Yeah.
So for the, sorry.
Yeah,
For
go
the
ahead.
sake of like growth standards, so reproducibility,
Mm-hmm.
I want to publish the data and all the processing that happened on this data,
Yeah,
leading to the figures that are on my paper,
yeah.
or whatever form it takes in
Yeah.
the future. How do I, provide that information, with all the down to the detail of the parameters that were used for?
Yep. Yeah, great question. Great question. This is something we're still trying to improve. We're improving. So the available way right now is not very streamlined, but that would mean publish your code. So the GitHub repository, you can maybe make a fork of that and then publish that. And then the data, the pipeline itself, there's two ways. You can publish the entire data pipeline. What I mean there is make the database server publicly available for access. That means you still have to host it yourself. So you're still paying some cost there, right? And then people can just get read-only access to your, but you can make a clone of that, and this is actually what we do quite a bit. You can make a clone of that that contain only the subject and the sessions for that publications. And then you put it somewhere and people can go to the code, and then they can link that to the actual pipeline. They would have probably read-only access, so they can only query things and check things. But if you grant them more access level, they can basically rerun the steps, right? They can delete the result and then rerun it. And with the same code base, with the same containerizations, the goal is they should be able to reproduce the exact same thing that you, all the way down to the final figures. Right? Because you capture everything in that pipeline already. But that's beautiful, but it's not quite there. Right. What we do today is we're trying to make better integration with Dandy Archive, but for Dandy Archives, we export into NWB and publish to Dandy is easy enough, right? We've done that many times and we know how that works. The problem there is we lose all of the intermediate compute steps. So when you publish to put things into NWB, you only store the results, right? Or maybe like the intermediate result, but you don't really store the code. So you cannot rerun it. So all you have is kind of like a...
Live results.
Yes, yeah. If you delete anything there, it's gone.
Yeah.
There's no way to recompute that.
Yeah.
So what we have to do is, what Dimitri is working on right now is a Dandy compatible DataJoint format that basically instead of
Correction, we're working on DataJoint compatible game.
Okay, sure. Yeah, wording matters. So yeah, the goal is to we can export data from DataJoint into files following the DataJoint, schema structure. So by itself, it's fully human readable and machine readable, but that itself can be, that's a set of files, but that itself can be kind of rehydrate back into a database itself, and then you can rerun the code, right? So, or it is a representation of the database. So we can then publish that onto Dandy as well. But that's kind of like work in progress still. Yeah. The biggest missing link right now is how to capture the code and how to rerun the code, because not just Dandy or NWB, but any other publishing or archiving or just data, right? There's no code there, so you cannot. So people typically publish the data, and then they write a repository with a bunch of notebooks that would say, read this data in and then run these things to reproduce the figures. That's like the most standard thing.
That's what everybody does.
That's what everybody's doing. So at time of publication, you have to go through and say, I gotta write all that code that would take my data format and then redo these steps and then make the figures that matches my figures in my publication. That's like, I don't know, like 6 weeks of work or something. So it's still, something.
Well, and actually the problem is also, it's like if you go to reuse someone else's code,
Yeah.
right, then it's hard to know where you should start.
Right.
Unless it's clearly written, saying, all right, first do this, then do this, then
Yeah.
do this, which will be basically a descriptive way of
Yeah,
doing
it's
the
like-
same thing here.
Right.
Here
For-
you have basically that whole thing, pipeline is like right there, right? Like this is a production pipeline. So
Yeah, exactly. This is production pipeline. So every time I don't know what's really happening here, I can just go back to the code and say, oh yeah, that's what happened there. And if the data is there, I can. And now you can actually ask AI agent to explain to you what's happening there. They do a pretty good job, like explaining, the low pass filter and then all that, specifically what happened there.
Guys, John, have you tried to see if you can also get a nurse shift to work with this?
No, we actually have not. That's something I wanted to try as well.
Given the fact that you want to do it like, a version that works with Dandy, I think that would be cool, because North is also doing a lot of stuff with the right? So it can just be like pulled up, then integrate this pipeline.
So that's coming. So we'll first meet with the Ember team, and then, because behavior is right now a big priority.
Yeah.
And then, but since Ember and Dandy are now very closely working together, one team together.
All
Yeah.
right, thank you very much.
Thanks everyone.
You guys are right.
No, this is great conversation. I mean, this is.